Start us out.
Great. Thank you guys so much for attending via Zoom. So this is a neat forum, and it's such a pleasure to introduce James Kardouni. At the time that I was kind of getting my head wrapped around the Elwha River Restoration Project, I was looking for a student that had a unique set of skills to work on this Elwha project, which is situated within this really complex watershed. And the project itself will intersect forest and river dynamics within restoration. And James really caught my eye, his experience with the natural resource, as a natural resource scientist from the Washington State Department of Ecology, and his work as a biological science technician from the Grand Tetons, and a long list of publications. I was really impressed. Situated him well as a watershed biologist, and I thought just perfect for this field site, which was difficult to get to. It was about a mile to 2 miles hike from the road into the plots. We got everything set up, and then in the big flood of 2017 washed out the road and created not a 1 to 2 mile hike into the field plots, a 5 to 6 mile hike. And I looked at James, I said, you know, you want to rethink this? We can set up another project. He said, no, forward. And I was like, right on. So that determination really shone through then in the field and continued on throughout the lab work and the writing. And it made this project for me such a pleasure to work with him and to get to spend the time in the Elwha River and also work with him on this thesis. Lastly, and then we'll get right to his presentation, I'm also so happy to welcome and congratulate him and his new position this spring as the water quality lead in Whatcom County with the Department of Ecology. So I'm very proud that he secured that job. I think that'll also intersect him with Western for years to come. So I'll turn it over to you, James. This has been such a fun project to be on, and I'm so pleased to have this time to hear your presentation.
Well, thank you, Janice. I couldn't have done it without you. Your expertise and skill set and creativity made the project what it is today. So appreciate that. And thanks everyone for tuning in. I appreciate Ed and Ingrid setting this up so we can reach a broader audience while we're sheltering in place. So I'll just get started and share my screen.
I gotta move a cat. Out, 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 out.
Okay, is everyone able to see my screen? So we'll talk about today my master's thesis, and we're considering forest restoration and how a particular flower here we see might affect forest restoration. Having a little delay on my slides. Okay. So here's an outline of what we'll talk about today. I apologize, my slides are really slow at flipping through. Okay. So the study area has some dams, had some dams on them, and they were removed, and it required some forest restoration. And so my research question is how lupine might be affecting that restoration. I'll go over the details of the study, our methods, our statistical approach, and that's kind of a schematic of some of the considerations we had during this project, ecological considerations, and we'll talk about the results, the implications of the study. So the study area is in Washington State on the Olympic Peninsula. So classic Pacific Northwest forest, and the watershed, the Elwha River watershed, drains mountainous terrain down to the valley floor northward to the Strait of Juan de Fuca in the Salish Sea. And there were two dams on the system, on the river system, and this yellow area is what I will be focusing on primarily, but I will just generally give you an idea of the impacts of the dams And we know that it did impact the sea-run fish, and it decimated them down. 90% of their population dropped. It impounded 90% of their habitat. And it also altered the riparian zone. The impoundments did, the dams did, as well as the upland forests through inundation. So after the dams were removed, it opened up a lot of protected habitat that's technically wilderness area. So it gave a great opportunity for the fish returns. However, there was issues to deal with, and one of them was forest restoration. And the forests, the health of the forests are related to fish. So after removal occurred, we were left with this 170 hectare exposed reservoir bed to deal with the revegetation. And that's where we're going to focus my study on. Revegetation, the forest restoration occurred almost right after drawdown. So they were in there, they knew it was important to do so. And some of the driving characteristics of the response of the restoration was partially driven by the sediment. So along the valley walls, we had very fine sediment and a different response in the vegetation as a result of that. And there were some other factors like water availability. And then where I'm going to focus is on these coarse-grained terraces. And the vegetation responded quite differently there. So now we're down on the ground and we're kind of where we were with the aerial photo. And we'll focus on the area between the valley wall on the periphery and in the floodplain of the Elwha River. And that's the coarse grained terraces, and that they get some effect from the river meander and such, but we're kind of out of that flood zone for the most part. And here's a close-up of one, or a look at one of these terraces. Here's Janice walking up in our study zone, study site, walking up the terraces, and you can see the coarse-grained, unconsolidated materials that the revegetation folks were working with, and we wanted to study that. And there's Janice, next to her is a lupine growing. So I'll talk about the forest restoration. Huge effort. Josh Chenoweth, he's part of the thesis committee. Unfortunately, he cannot be here today, but he headed up the restoration, and they realized they needed to establish plants as soon as possible. And the reason, so there's a lot of reason for it, but just to regain that forest functionality to support the ecosystem and the riverine habitats as well. They planted over 300,000 native trees and shrubs from the greenhouse. They also seeded pure live seed mix of a variety of seeds, of locally sourced seeds, and 2 of which were lupine. Lupine polyphyllus, which is big leaf lupine, and Lupine rivularis. And there's a nice little picture of one just emerging from those cobbles. And you can see a seed pod there next to it as well. So there were some challenges in the restoration efforts. And here's Janice in our study area, and we can see, again, that coarse, unconsolidated material, the sediment that people were working with when they were trying to re-veg. And the soils were very limited in resources such as nitrogen. And it was so far away from the intact forest, there was limited legacy resources to help establish that. There was limited water availability because of the infiltration rate. The water could percolate through those unconsolidated sediments very quickly. And also wind and temperature effects. Massive evapotranspiration rates led to high mortalities, and invasive species can exploit these harsh conditions. And then there was also species competition And a lot of that's driven by site availability, the species that are present, and how they perform. So there were some challenges in the area. And as a result, we see that the restoration varied. So I'll just talk about where we're not studying. The valley walls established rather quickly with that fine sediment. And there's a picture of Josh trudging through a thicket of, looks like alder, or maybe some poplar. And it got thicker than that. If we took a photo of him in the thicket of it all, we probably wouldn't see him in his green uniform. So what we're gonna focus on today is that valley bottom where there's coarse sediment. The vegetation really struggled to get there. However, one star of the show was the lupine. And of the 2 lupine seeded, it was riverbank lupine that really took off. And there's a photo of Josh in the first cycle of flowering stage of the riverbank lupine shortly after the seeding. So you can see it's really starting to get established. And here's a couple photos of lupine in its vegetative state and in its blooming state. So we'll talk about lupine ecology and how that might serve to work into the system. It is a biennial, sometimes I don't know if triennial is the actual term for it, but it might have a 3-year life cycle essentially. And we know it's nitrogen fixing through rhizobium. It lives in the root nodules of the lupine, and nitrogen is essential, especially to young Pacific Northwest forests. It's an essential nutrient, and young forests are often limited in nitrogen. The lupine also helps with soil development by producing organic matter as it dies off naturally, and it probably holds moisture content and aids in other nutrient cycling in the system. It also tolerates harsh conditions like those coarse materials on the terraces here. And we're near the anniversary of the Mount St. Helens eruption. That picture there is lupine lepidus, Pacific lupine, growing on the flanks, the pumice plains on Mount St. Helens. And that is a very harsh climate. They're essentially sterile soils that lupine came in and established there early and helped pave the way for other plants to establish. So a lot of my studies based on what folks found there. And here's a photo of the Elwha. This is one of Janice's study sites, and we see our re-veg conifers there in the foreground. In the background, lupine getting started in their first year of growth. And so we're curious, as lupine establishes to a more bigger biomass, a larger biomass, is it really helping, or is it dominating? Is it inhibiting forest recovery? And the conifers are. So that's our question. And so that leads me to the purpose is we want to develop an understanding of lupine in managed ecosystem restoration. So we're going to investigate interactions and what lupine might provide to the system. And so we identified some objectives to measure by doing so. And my slides are freezing again. Let's see. Whoop. Okay, so here we see a photo of a little lupine sprouting out, germinating in the base of one of the restoration conifers. And I hypothesize that it will benefit forest recovery. And we also, part of the benefit will be an increase in tree growth and plant recruitment, as well as ectomycorrhizal colonization. And I'll talk about a little bit about ectomycorrhizae for those that don't know much about them, or for those that do as well. But we think that we can assess these indicators of restoration. So here's an aerial ortho of the Lake Mills Basin. It's about 170 hectares. Those yellow dots are our study sites. It's in the managed zone on those coarse terraces above the floodplain in between the valley wall. We established around 24 larger treatment areas, and I'll talk about those in a bit. and a total of 72 sampling points. And so as we established our study sites, we wanted to, we gave a general idea, what's the lupine doing here? How can we categorize it into certain levels? And we came up with 3 levels, the sparse, medium, and dense. And these are photos. There's Janice in the dense. You can see it's waist high. It's quite well established. And we see the photo in the middle of medium where there's some lupine in there, but not as much. And then the sparse where there's very little to no lupine. And our measurement objectives to assess the restoration process, we looked at conifer growth, the plant species assemblage, soil organic matter, and the soil nitrogen levels and the foliar nitrogen levels in the re-veg conifers. We looked at the ectomycorrhizal colonization in the root tips of those conifers. And we also wanted to account for some random factors that might be driving the successional response, the recovery of the forest. So we considered tree species and soil texture, essentially the sediment particle size. So here's an example of one of our study plots. And it was essentially the centroid of the clump. It was an 8 by 8 meter square. And after assessing the area, we came up with these notions of a dense plot. So we would go to a dense plot, have an 8 by 8 meter square, and tally up all the conifers. And then we'd randomly select 3 conifers, and those represent our sampling points. And around each conifer, we have these circular quadrats that we could assess the vegetation and also assess for random variables like the sediment texture. And so I'm going to go through each measurement variable and what we did. So these are some photos of the conifers, trees we selected. And we ended up with 63 total sampling points because some of the conifers died. And our study started about 3 years or so after, maybe 4, after the restoration, the initial restoration in the Lake Mills Basin occurred. So it gave time for some of those trees to die even after we selected them the previous year. So we measured the tree root collar diameter and the tree height, the foliar nitrogen, and grinding the foliage, it smells amazing. But we ground it to a powder, ran it through an analyzer, got that. We also got the tree health index, the vitality, how green the leaves were and things like that. And then again, the ectomycorrhizae on the root tips, the colonization percent. And here's an example of our vegetation survey. That's me working. We're assessing, we had 63 plots around each conifer. The photo on the left is a conifer in the middle, and then there's that kind of yellow outline of essentially 2 meter square plot. And we identified all the plants in there, and then we assigned them a cover class, 1 of 7 cover classes. That's their percent cover. And then our soil characteristics methodologies, we trenched some soils to get the moisture content, the organic matter content, and how much nitrogen was in the soil. And then we also assessed for the random factor of the soil particle, the sediment particle size. So we did a number of counts within that study quadrat and perpendicular lines. So we total about 36 counts per quadrat. And now a little bit about the ectomycorrhizae. The ecology, here's a couple of photos. So what they are, they're a symbiont between trees and fungi. And they form in the interstitial cells of the tree on the root tips of the trees. And this is a photo of Janice collecting some of the samples. She gently took some root tip samples of the restoration conifers. And we think that the ectomycorrhizae are an important indicator of healthy mature forests, they comprise a huge part of the biomass in mature Pacific Northwest soils. So they are essential. They access nutrients and water for the tree, and the tree in turn gives the fungi carbon and resources to grow and even reproduce in some cases. And it's through that interface of the ectomycorrhizae that this occurs. So anyways, what we did is we collected some root segments, and we wanted to know the colonization rate. So one way to do it is to look at the morphotype, so essentially the physical presentation. And these are photos of ectomycorrhizae in root tips of trees. And we also backed that up with the DNA sequencing of the fungi associated with those morphotypes. And then we quantified how much occurrence occurred on those root tips, how much ECM were on the root tips. So that concludes the methods, and then part of the analysis of what we measured, we used univariate analysis, and a mixed effects model was an appropriate choice for this study. And this allowed us to confirm a response variable. Here's a schematic of the model. I'll just go through each term. So a response variable is on the left there of the equation. That is the dependent variable in the model, and it could be foliar nitrogen, for example. And then our fixed effect in the model is the lupine treatment levels, such as dense, medium, or sparse. And then the next term is S there. That is a random term in the model, and that is the median soil particle size of our pebble counts when we did our surveys in the quadrats. And then the other random variable was what tree species. We ended up randomly selecting within our study plots 3 species: Doug fir, grand fir, and white pine. And maybe their species determine some physiological response in the system. So we made that a random variable, and it was nested in treatment, the dense, medium, sparse treatment. And then we have our other terms in the model. And then our second statistical approach was a bivariate analysis using correlation, so we can see how variables relate to one another by doing so. And then finally, we look at all the variables collectively in the system. And one great way to do that is through principal components analysis. You can distill and see how, and then followed by cluster analysis, you can see how sampling points, sampling sites might be similar or different from one another. So we'll get into the details. But first, I'd like to confirm we had our predetermined treatment levels through visual delineation, and we stacked up pretty well. Here we see that this is a density plot of all 3 of our treatments, and the mean value comes in at roughly 76% cover, the median at about 58, and the sparse at around 15. So we're not bad. We're in the first and second, third quartiles of the distribution. So I'm happy with that. And we'll start off with the model results. And what we did with the model is we wanted to test for significance concerning the treatment and the random factors. We didn't see that the random terms in the model, the random factors had no bearing on the outcome. But we did find, using an ANOVA test, that there was some bearing on the outcome, that there was a significance based on treatment. And I'll talk about each one of these graphs here. And then we followed up with a pairwise comparison to where we can see differences among the treatments. So we can really fine tune and examine it on a closer level. So let's start with the top left there, plot A, and this concerns tree growth. And what we see is that the medium treatment we had significantly larger trees in the medium treatment plots than the sparse. However, the dense treatment was similar in stature to the medium and the sparse. And the predict up there, it's 45%. So what that's telling us is model fitness. So the model was able to predict this outcome, and we ran 1,000 simulations of the model to predict what the distribution of our actually observed data. And that's what we're seeing here. The salmon color is what we actually observed, and then the teal color is what the model predicted. And this model was about 45% of the time was able to land in that interquartile range of our actual observed measurements. So that's a pretty good model. But the take-home message is mediums, plots, those trees are growing larger than the sparse. And let's go to B here. This is concerning the percent of the concentration of foliar nitrogen in those restoration conifers. And what we see is that the dense and medium treatments are significantly higher in foliar nitrogen than the sparse. That's good to see this play out in the model. And on top of that is the model is the fitness is great. That it could 85% of the time through simulation predict. So that's a decent model. Now we're going to move to C here with ectomycorrhizae. What we found out is that there were more ectomycorrhizae colonizing. This is a percent colonization on the root tips. At just over 30% roughly, we saw a colonization rate in the sparse. And that was significantly different than the other two treatments, the dense and medium treatments of lupine abundance. And this model performed really well, that 95% of the time, the simulated outputs would fall within that distribution of the observed. So that's a good one to see. And then finally, plant species richness, we saw that the sparse plots had a higher richness, so a higher number of plants when compared to the medium. However, there was no difference between the dense and the sparse and the dense and the medium. The model predictive power was not so good, so the model is not very fit. If 3% of the time it was able to come up with the somewhere within the range of the observed. All right, so that's our model results. I want to talk a little bit about the fungi that we did see and the ectomycorrhizae they form. Again, it was we didn't really see that much. At most, we saw 30% in the sparse. And so the first two here, Telephora and Wilcoxina, they are common in greenhouses. So we think, and it was mostly the percent abundance of all the species that we did count. Telephora is telling us that it represented 56% of the total population that we saw of all the fungal species. So we suspect these may have just come in on the greenhouse because they are common. And then the next three here, these are pioneering species in the Pacific Northwest, and they're just getting started. As we can see, they're not very abundant. And then the last three here, they're not ectomycorrhizal. We did get that in our genetic signature. So we picked them up. But they are fungi. Now we'll look at how variables interact with one another, what their relationships are like. So here we'll start with the bottom row with lupine cover, and we see that there's a positive correlation between lupine cover and foliar nitrogen. And this kind of confirms what we saw in the model. Second, we look at the lupine cover and our ectomycorrhizal colonization. And this again confirms what we saw in the model, that there's a negative correlation. And tree growth as well. We saw this in our mixed effects model with a positive correlation between lupine cover and tree growth. And a couple other things to point out is that lupine cover had a negative relationship. So the more lupine, the narrow the root collar diameter of the tree. And what we also saw in the mix effects model to some degree, that lupine cover had some determining influence on the species richness, the surrounding vegetation species richness. And here we see that negative relationship. And I'll talk a little bit about the trees, and we'll hang on to this thought here, but the tree diameter, we saw larger root collar diameters where there was less nitrogen. So there's a negative relationship between root collar diameter and foliar nitrogen. And I'll talk about the implications of this in our discussion. And here, kind of on the same train of thought, is where we see a larger root collar diameter where there was more ectomycorrhizae. And then our tree metric was essentially our tree growth metric. I just wanted to assess that. We got a pretty good relationship between tree diameter and the root collar diameter of the tree and the tree height. That's good to confirm that. And then what was driving that metric was tree height primarily. So we'll close up our results here with the multivariate analysis. And I'll explain this plot, take some time on it here, but we omitted lupine cover from this analysis. So it's kind of like a blind or unsupervised classification of the system. And we've considered certain variables and how, so what this is telling us is how the sampling points, how they are similar to one another or different, and what's driving those similarities. Whether it's foliar nitrogen or species richness. So this tells us how these groups were similar or different and why. So let's start with cluster group 1. It's the orange cluster group. And so after the principal components analysis, you cluster on the principal components. And this was the majority. I think it was over 50% of our samples landed in this. And the breakdown is that most of them were medium, followed by some dense and sparse. So all these groups were considered similar. Their variables showed some similarities. And this ordination plot shows them kind of grouping together. And the cluster analysis shows how they clustered together with hierarchical clustering. And what we see, what's driving their differences, or this particular group's similarities, is that species diversity was one of the biggest. It's a relative to other variables. Species diversity was driving it, substrate, and then foliar nitrogen and tree growth. So these groups were similar with respects to those variables. And second, let's look at the green group here. That's the lower part of this ordination plot. And what we see, it's mostly dense. And what's driving it? Foliar nitrogen. So where there's more lupine, there's more foliar nitrogen. So it's a good way to confirm that and see that kind of play out in this ordination plot. And then our third group, the red group, was primarily our sparse treatments. And they ordinated on the left of this ordination plot. And mostly sparse in there, and what's driving that was the bare ground. So that's where we didn't see vegetation, we included that. And then the ectomycorrhizae and species richness. And those two are pretty important, as those variables are lending themselves to group these particular sampling points together. So those were the results. And we can now talk about the implications of what we found. And my take-home message is that we are finding supportive evidence of lupine's ability to help with restoration in this highly disturbed restoration site. You can see on the left, that picture on the left, there's a high degree, there's mortality going on. not a lot of soils to work with. So these lupine might be helping this restoration process. And how's it doing that? So we saw that there was larger tree growth with more lupine. And what might be allowing that to happen is that the lupine is providing that nitrogen to the tree. And there's evidence to show that increased nitrogen in leaf biomass increases photosynthetic efficiency. So that's probably what we're seeing play out here. When that lupine makes that nitrogen available, it's growing larger as a result. And other studies have shown that in the first 20 years of restoration, accelerated tree growth does happen with lupine treatment. And that was on a completely bulldozed site. So they were working with very little legacy resources. So we did see an enhancement of tree growth, and we didn't really see any encroachment of overcrowding, but we really don't know if there's an optimal seeding density for lupine. If you're going to include that in the mix as a restoration practitioner, that's something that we could take the notes of what Josh came up with and come up with that idea of the optimal density. And now let's talk about vegetation cover. So how lupine affects that. And what we see is that lupine correlates with the vegetation recruitment, and in particular where we think lupine might serve as a cover crop because we saw less species, so lower species richness where there's more lupine. And what we kind of distilled our species list, and what we saw is that we saw more invasive grasses where there was less lupine. All the other species we couldn't really safely say that there was more of or not, but in particular, those invasive grasses were present. So that makes this is evidence, supportive evidence of lupine acting as a cover crop and helping out the system by preventing those invasive species from occurring. And now about the ectomycorrhizae, what we saw was, in general, not a lot of colonization present, at most 30%. And that happened on the sparse treatments where there was not a lot of lupine. And what was interesting, of the taxa we found, we did not see rhizopogon. And rhizopogon is an essential and important ectomycorrhizal or fungi that forms an ectomycorrhizal symbiont in Pacific Northwest forests. So this is indicative of we're just getting started here, and the forest restorations, that it takes a while for these fungi to start coming in and showing up in the system. And other studies have shown that Cortese and Bunn have studied abruscular mycorrhizae in the system, and they saw that there was not a lot coming into the system, and there might be some forest edge effects where the closer you are to the forest, the more you're seeing, the more fungi are present. And fungi are really important for forest ecosystems. They have a huge, huge service in there. So we're seeing this ectomycorrhizal, this inverse relationship, and we're not quite sure what's going on, but we think that lupine relaxes the necessity for that ectomycorrhizal symbiont. And so one indicator of below ground resource allocation for a tree is the root collar diameter. And remember, we saw a larger root collar diameter in the sparse, where there was less lupine, right, in the sparse treatments. And so we think that maybe the trees are adopting those symbionts and putting more resources underground, and it might be showing in that root collar diameter. So here, maybe it's resource based as to why if there might be less nitrogen in the sparse treatments, then those trees are resorting to the ectomycorrhizal symbiont to access what is limited in nutrients, because we know that the ectomycorrhizae are essential at getting at those resources for the tree and such. And then a discussion about soils. We were curious how lupine might affect soils, and we did not see that there was an increased levels of organic matter in our soil samples. That picture on the right does show promise. I think we're a little too early to detect it, because you can see as the lupine, as a biennial species, it drops its leaves, and that's cycling organic matter back into the system and getting it ready to start, you know, keeping that organic matter that's essential for soil development. So we think that there's some potential. Didn't detect it. And then another one is the soil nitrogen levels. We didn't really see elevated nitrogen levels where there was more lupine. And we think that either the plants are assimilating it right away, or it's leaching out of the system, because this system nitrogen is pretty mobile, especially when there's not a lot of organic matter in the system. And so we do have some future questions. And we want to know what lupine's long-term influence is on forest recovery. Again, we went in with our surveys and did our study about 3 years after the initial restoration occurred on these terraces of the Lake Mills Basin. So there's definitely, we can see the future successional patterns play out, and we can consider lupine's function in managed restoration and its importance. So we would like to see more studies concerning the soil organic matter and nitrogen levels, as we think that lupine might affect those in the long term. We also think that lupine provides some type of microclimate, and part of that is a soil moisture retention. We didn't have a sophisticated way of assessing that. Yeah, we dried our soils, we didn't see a difference, but there's more sophisticated ways of detecting those microclimates that lupine might be helping with forest restoration. We also want to know more about the fungal interface, the ectomycorrhizae, and I didn't talk much about abruscular mycorrhizae, but we know some lupine does associate with abruscular mycorrhizae. We didn't assess that for our study, because we were assessing the conifer trees, but maybe that could be a key to lupine's success in growing in these harsh environments. And we also know that abruscular mycorrhizae can form a symbiont with Doug fir. So Doug fir can form symbionts with ectomycorrhizae and abruscular. And that would be an interesting question to answer as well. We didn't assess for abruscular mycorrhizae. Where is it in the system? We also want to know about long-term tree growth as well. Is it going to assist further on down the road? Some studies have shown that about 20 years after lupine treatment, the trees don't show an accelerated growth because of lupine treatment. And we also want to know a little bit more about its ability to serve as a cover crop. And there's some possibilities. We're starting to see some promise here with this study. And then long-term lupine abundance. We know we saw photos of it really dominating in some pockets here and there, but what's going to happen before we see canopy closure? Will it continue to dominate? Will it continue to help or not? And then under canopy closure, will the lupine start to grow less dense, and nitrogen fixing is very energy intensive, so you need a lot of sunlight to do so. And so we would like to see, as these forests grow tall and the canopy closure occurs, what happens to the lupine. But I do think for now it seems to be benefiting the forest recovery efforts here. And I appreciate you all listening. And here's my thesis committee, Janice Bauman and Robin Matthews, Jim Helfield, and Josh Chenoweth. They've all provided great feedback, and I appreciate your help. Also, funding was provided externally through the Stunts Foundation and internally from Western, the research sponsored program, and the Ross Travel Grant. So thank you all. I guess there's time for questions, and I'm not sure how that works with audience members beyond the committee. If Ingrid knows, or Ed.
I think we might have some time for a couple questions.
Okay.
So any questions for James?
James,
Yes.
I have a question about your ordination. Can you go back to that slide? And I might have missed it when you introduced it, but my question is basically which data are in the ordination? Is it so for each of these points on your ordination, which data are the multivariate response? Does that include bare ground richness, ectomycorrhizal colonization?
Yes, essentially, yeah, all the variables that you see on the plot were included in the ordination. And those arrows show their relative magnitude of the influence on the ordination.
And then, so I think, did these arrows then, was this done in R?
I tried, and I had to doctor it up in PowerPoint.
Okay, that's fine. Yeah. It's unusual to show the variables that are part of the ordination regressed back onto the ordination. So it'd be more common maybe to show the loadings for the principal components. I don't think it won't change your interpretation at all, but it's just a more common way to present that. I can follow up with you guys later. I just wasn't sure when you were explaining it. Okay,
yeah, and so some of those bullet points, ordination, so let's just take the first, or if you can see my pointer, yep. Here we're seeing that the loading score, or the loading, the strongest relative loading was species diversity, followed by substrate. And I probably didn't explain that very well.
Yeah. Okay.
I mostly focused on the separation and what was driving it.
Let me ask a question, because I'm assuming this is an R biplot, so that you're actually plotting both the sample ordination as well as the variable loading on the same plot. Is that correct?
That is correct.
Okay, that's what I think. So you're actually seeing those biplots, you're actually seeing the variable loadings on there where the end of the arrow is.
Yes.
But then I had a follow-up question. I want to make sure you finish Rebecca's question first, but I have a follow-up.
I think that's good for now. That helped me out. Thank you, James.
Yeah, I'd be happy to talk shop with you. I like this kind of stuff. So yeah.
That sounds good.
Well, so my question is that looking at the green cluster, you made a comment about it being driven primarily by foliar nitrogen. You want to explain why you would say that it's driven by foliar nitrogen as opposed to, say, foliar nitrogen and richness, since those would have the same basic score on principal component 2.
Oh, yeah. I overlooked that, and I could.
Okay.
So yeah, for that particular group, I could see what's driving that, and maybe richness lines up.
Yep.
Right behind foliar nitrogen.
Yeah, you could, because you can see on PC1, they're all in line on PC1. You just drop them straight down. There's no separation to speak of on PC1. The separation occurs on PC2. So just, I don't know, might want to rephrase that or something.
Okay, sounds good.
Can I ask one more question?
Yes.
So I want to zoom back out. That was really specific. I'm sorry, James. I should have started with great presentation, interesting project.
Thank you.
But what I'm curious about, one of the things that I wondered that I can't remember, when they seeded the restoration sites, was lupin in all the seeds, and was it distributed evenly across all the sites? My question is really, was there any difference in seeding that might have explained some of the differences in density of lupin across the substrates?
Right. I suspect the same as well. And Josh could answer the question better, but what we do know is that it was an arbitrary seed mix. So the thing that was constant was the amount of seed per square meter. So that density was constant. But as far as what seeds were in that mix, that was arbitrary. And maybe we're seeing that pattern play out here, and it's too early to really tell.
Yeah. Okay, thank you.
Yeah. Good question.
James, I don't know if you guys are monitoring the group chat, but looks like Carolyn had a question for you.
Could you moderate that for me, please, Ed? Because I really,
Sure.
I'm afraid to exit out and have things just go haywire.
All right. Well, Carolyn says, super interesting project. Was the revegetation done in such a way that those vast swaths of lupine early on were to be expected, or was the lupine extremely successful simply because they can fix their own nitrogen and were better suited for the harsh conditions? And then she goes on, when choosing the high, medium, and low lupine density plots, was that based on how the revegetation was done, or were they naturally occurring densities?
Well, I can't say whether it was naturally occurring because of the question that Rebecca brought up about lupine density. What is natural is that they can really thrive in this harsh environment. So we just took that into account, and regardless of if the seeding was consistent across the board, we just wanted to know if lupine was really causing a difference in the vegetative assemblage.
I add a little bit to that, James, just because before you came, Josh and I were touring these sites. And just to add a little more to answer both those questions, yes, lupin was arbitrarily mixed in. So some of these areas just got more lupin seed during the seeding event than others. And unfortunately, those notes weren't documented very well. And that we need to follow up on that to find that ideal seeding. Another thing before James got there was we were like, wow, this is really neat. The lupin are doing really well. And then the offspring of that initial seeding went crazy. And that, I believe, exceeded expectations of lupin's ability to really come in very, very thick. So they knew, thinking of that Mount Rainier, Mount St. Helens project, that lupin will probably do very well. So that's why it was included. But the fact that it not only established, but then started to seed that next generation, I think exceeded the expectations.
I'm not sure I answered Carolyn's question, but I can follow up with her if she likes.
Okay, I think we have time for maybe